The overall goal of this procedure is to determine the genetic control of translocation formation by single-strand annealing in diploid budding yeast cells. This is accomplished by first inoculating cultures with single colonies of a specific genotype and growing them overnight. The second step of the procedure is to induce simultaneous double-strand break formation by HO-Endonuclease on 2 chromosomes by the addition of galactose to the cultures. Next, dilutions of these cultures are plated on selective and non-selective media. The final step is to determine translocation frequency by counting colonies arising on selective and non-selective media. Ultimately, results can be obtained that show genetic control of translocation formation by single-strand annealing through changes in the frequency of HO-stimulated translocation formation in strains of different genotypes.
The implications of this technique extend toward the consequences of therapeutic radiation exposure of cancer patients, because DNA double-strand breaks and translocation formation are the primary consequences of this treatment.
To begin the procedure for determining HO-stimulated translocation frequencies, inoculate 10 to 20 independent 1 milliliter cultures of YP glycerol lactate medium with single colonies of the desired genotype. Incubate the cultures overnight at 30 degrees Celsius on a rotator. When the cultures have reached the desired cell density, add 20% galactose to the cultures to a final concentration of 2%. The galactose will induce expression of the HO-Endonuclease, which will direct double-strand breaks at the his3 delta 5' allele on chromosome 3 and his3 delta 3' allele on chromosome 15. Incubate for 4 hours.